This is a production of Cornell University.
All right, thanks everyone for taking the time to be here. Today I'm excited to share my dissertation work titled How Do We Know a Soil is Healthy and How Does It Relate to Soil Carbon Sequestration? Just for context, I want to share what my whole dissertation involves, 5 chapters. Today I'm not going to be presenting on chapter 1, cropping system and soil texture shape soil health outcomes, and chapter 3 on the development of pedotransfer functions for available water capacity, because I presented on those topics 3 years ago in department seminar. But today we'll be focusing on chapter 2 on production, environment, soil health benchmarks, considering soil type, cropping system, and regional context. On chapter 4 on the development of pedotransfer functions for soil protein, and on chapter 5 on the effects of tillage, cropping system, and soil type on soil organic carbon stocks at 3 of New York's oldest ag experiments. So to set the stage for today, I want to discuss the triple threat to agriculture and global food security. Seen from space, humans have been a soil degrading species. In the last 30 years, 22% of agricultural land has undergone measurable soil degradation. And here we can see wind erosion taking soil particles away, but we don't see the invisible flux of carbon dioxide leaving the soil. So if we don't replace the organic matter that's lost each year, we'll have soil degradation, loss of soil functioning. At the same time, we have agricultural nutrient pollution that exacerbates global warming in the terms of nitrous oxide and threatens aquatic water bodies globally. Meanwhile, we have extreme precipitation events, droughts, heat waves that are predicted to increase in magnitude and frequency. And this farmer right here is from 2016, where there was a 100-year drought in Western New York. And these events are predicted to increase in frequency. And this triple threat really sets the stage for the importance of sustainable soil management strategies that increase carbon inputs and reduce disturbance to increase resiliency and potentially mitigate some of the impacts of climate change. So it's not all doom and gloom. New York is a leader in soil health and climate change with some of the most ambitious climate legislation in the country. New York signed the Climate Leadership and Community Protection Act in 2019, also known as the CLCPA, which calls for 85% reduction in greenhouse gas emissions by 2050. So incredibly ambitious target. At the same time, we also have some very exciting soil health and climate resiliency legislation that was passed unanimously in December 2021 and calls for increase in financial and technical assistance for support practices that improve soil health, increase climate resiliency, and reduce greenhouse gas emissions, a commitment to scientific research on soil health and carbon sequestration, and the establishment of voluntary soil health targets, which is a perfect segue into my chapter 2, which is about developing the foundation for this science. So chapter 2 is about production environment soil health benchmarks that consider soil type, cropping system, and regional context. And this is really the scientific foundation that we're proposing could be the foundation for these voluntary soil standards. Just some terminology introduction. Production environment might be a new concept, but that word really helps to encapsulate the soil type, cropping system, and regional differences in a single peer group. So we could, an example could be loam textured soils, annual grain cropping systems in upstate New York. So just for some context, our lab group uses the Cornell Soil Health Laboratory, Comprehensive Assessment of Soil Health. It's the first commercial soil health laboratory in the world. And the CASH test captures all important soil processes, physical, biological, and chemical. And there's 4 physical indicators, 4 biological, and 4 chemical with their measurements. And then the CASH framework interprets those measured values based on texture to give ratings that help interpret those measure values, helps to identify soil constraints, provides guidance for management. And the lab has processed a huge number of soil samples over the last 10 years and been a real model for this comprehensive soil testing approach and been a major tool by different research and outreach initiatives across the country. So we can think of three major factors of soil health, and you might have seen this conceptualization from Valentina Rubio. We can think of the inherent factors, things like parent material or mineralogy, climate, biology, slope, and time. And these factors all come together to shape inherent soil properties, things like texture, drainage. Those are properties that we are stuck with and have no control over. Then there's cropping system. These are the cropping systems across the landscape. In New York, we have an incredible diversity of cropping systems, annual grain, orchard, vegetable, pasture. And this really impacts soil health through the different carbon and nitrogen cycling regimes that these different systems have. And because some of this is structural, there's limited control in changing the cropping system. Because if you're growing annual grain, you're not likely to switch to a pasture system, for example. And this is another good opportunity to introduce this production environment concept. This production environment really is creating a peer group of inherent properties and cropping system that we can use to create benchmarks and interpret soil health in the context of natural soil forming factors and the cropping system context. And finally, within each of those cropping systems, the farmer has ultimate control to choose their crop rotation, cover cropping, tillage choices, organic amendment choices, fertility choices. So within each of those cropping systems, there's that third level of the management specificity within each cropping system.
So first, I'm going to talk about these factors of soil health, these inherent soil types, and also the influence of climate on soil health measurements. And we really need to get a handle on these inherent effects so that we can assess the effects of management. So here's biological indicators by soil texture. So we have 4 soil texture groups and 4 biological indicators that are included. And we've known as soil scientists for a long time that finer textured soils with more fine silt and clay tend to have higher organic matter and stabilized organic matter against decomposition than coarser textured soils. The same is true for some of these other more novel biological indicators that are more sensitive to management. And this is really important for being able to assess the effects of management. And a good example is, let's say you're a sandy loam soil or a coarse textured soil. Having an organic matter of 4.1 would be really, really good for you, but would be a pretty average value for if your soil texture is silt loam. So really important inherent context for interpreting soil health. Next, when we can think of the regional impacts on soil health, and we split it out by region, but I really want you to think about climate. One of the regions that really pulled apart as we were doing this New York soil health characterization work was kind of the inherent differences between Long Island and the rest of New York State. So it appeared when we were comparing all these the same cropping systems and the same textures between the rest of New York State and Long Island, we were seeing large differences in organic matter between the two. So here we have, these are all just pastures. So the coarse Long Island had about 1 to 2% organic matter differences across these whole groups. And this makes sense from a climate perspective. This makes sense when you talk, or resonates when you talk to people like Olga Vargas, who work on Long Island and in other parts of New York, who like know these soil color differences and know this is true. So Long Island has a lot more coarser textured soils. It's kind of the glacial moraine, and also has quite a bit warmer climate, maritime climate compared to the continental climate. So this is really important context to think about. We also know that cropping system and the degree of nutrient cycling, disturbance, carbon balance influence soil health and all those different management practices. And you can think of 2 extremes in terms of cropping systems and their influence on carbon and nutrient cycling and balances and degrees of disturbance with this example. So we have these natural or pasture systems, photosynthesizing year round. 90 to 100% of that biomass is cycled in place, doesn't leave the site. We have no disturbance through tillage, and happy earthworms. And then we can contrast it with a system that we're using really intensely for agriculture as an annual grain system or a processing vegetable system, where we're harvesting maybe 50 to 70% of the carbon and nutrients in our crop harvest and export. We're having other losses from tillage and erosion, and less cycling of organic matter and less happy earthworms. And in conventional and traditional agriculture, we replace the nutrients with synthetic fertilizer. It's easier to move around, lighter weight, but not that carbon, which leads to soil degradation. So think about this in the context of the next results I'm going to give you. For the past 5 years, we've been really fine-tuning and looking at the effects of the different cropping systems in New York on soil health. So now we're looking at 6 cropping systems. We have annual grain systems, corn, soybeans, sometimes corn, soybean, wheat rotations, maybe dry beans, growing the grain. We have a lot of dairy. Dairy's really big in New York State. Corn silage, alfalfa rotations, but there's a trend towards rotations that have more corn silage in them. We have a lot of pastured dairy and beef in New York State, continuous living roots all year round, orchard systems, perennial systems, so some heterogeneity within the row and between the row. So I leave that complexity to Debbie. We have this mixed vegetable system, diverse array of vegetables grown in a smaller area. These tend to be kind of your organic vegetable farms, market gardens, growing a diverse array of products from market. And then mostly in Western New York, we have some really larger processing vegetable farms growing tens to hundreds acres of cabbage, snap beans, pumpkins, sweet corn, etc. So this distinguishment is important. So here we're going to look at soil organic matter and aggregate stability in a second by cropping system. And we're controlling the inherent context because we know that those things really affect soil health as well. So we want to be able to compare apples to apples. Orchard in there too. And we know that we were seeing consistently across soil texture groups that the pastures and mixed vegetable operations maintain the highest soil organic matter. You can think of dairy as being kind of intermediate, has this very intense part of the rotation with corn silage, but also has a lot of manure and perennial forages. And then our systems that are most intensely used, annual grain and processing vegetable, have the lowest soil health outcomes. We're using them very hard for agriculture. For aggregate stability, really important physical soil health indicator about soil structure and soil resiliency. Pastures had 2 to 3 times as much aggregate stability as all these other annual systems. So it has a really important signature of disturbance or lack thereof disturbance, and really resonates with all the work that's been done that shows that continuous living roots and their associated mycorrhizal fungi really help to build and maintain aggregates. So we have these this growing New York State database with soil type, region, and cropping system context. So now we're working to advance the interpretation of soil values so that farmers can compare themselves to their peers, which is who they really want to compare themselves with. So here's a real dairy farmer I worked with. I obscured his name, so Moose Dairy Farm. We have some soil health benchmarking graphics for the farmer. And this was really nice proof of concept for him. Been really invested heavily in no-till and cover crops for the last 15 years. So a really nice proof of concept that his organic matter was in the top 20th percentile and one of the higher aggregate stability values we saw in that peer group. And we've also been working on soil health benchmarking infographics for the consumer. So these would be for farms that are more consumer facing, small scale market gardens, orchards, farmers that are interfacing with the consumer. So this might be a tool that could be placed on a website. And actually, if you check out Norwich Meadows website, they just updated it and have their first feature. They were kind of the pilot farm for this development. Last year, or you might have this printed out in a poster for farmers market, and you could share this with buyers. So this is made to be simple, just with a soil health score, and then we focused on carbon instead of organic matter because we know that there's this huge growing interest in the public for carbon farming and climate change mitigation and all that. So setting benchmarks for soil health, benchmarks exist for water and air, so why not soil? Soil health benchmarks can help farmers calibrate their management more specifically and provide kind of realistic targets compared to their peers. And they can also potentially be used in different policy initiatives. So, but as we showed, these benchmarks need to be adapted to soil type, region, and cropping system context. So this is what we're calling our scientific foundation for these benchmarks that will be handed over to the state to develop these voluntary standards. So we've calling them production environment soil health benchmarks, or PESH. And an example again, because this production environment concept is maybe not straightforward, is that a production environment might be an annual grain, loam textured soils in upstate New York. So we have annual grain and pasture here. And this is based on kind of a probabilistic or fuzzy logic approach. And this is similar to how blood pressure is interpreted by weight or size or something like that. So we've decided the 90th percentile is a good way to start the conversation. It's a realistic goal within the peer group. 10% of the farmers' fields have been able to achieve those goals, and establishes clear pathways and realistic targets. So if you start with an organic matter value of 2.5, you know that it's possible to achieve this 3.7. We also thought it was really important to develop a universal benchmark for all of the different cropping systems, and pastures are that kind of best case scenario for soil health. So for a given soil type or region, kind of represents that upper limit. In the past, we've always, with our soil sampling, we've always recommended contrast sampling. So now we have that kind of pasture data to assess the effects of our management in other systems. And in our lab, we focus a lot on extension materials, and these are some of the extension materials that we've developed to share this with the public. And here's a new hot off the press soil health benchmarks for New York's farm policy brief that just came out. All right, so now I'm going to transition to chapter 4 on pedo transfer functions for soil protein based on random forest modeling for routine soil health analysis. Basically, the idea of replacing a measurement with a prediction for more cost-effective soil health assessment. In terms of terminology, a pedo transfer function is basically a model that helps us predict something that's costly to measure from something that's easier to measure. So background, soil protein is an important biological soil health indicator, provides an assessment of the size of the organic nitrogen pool, and provides an indirect measure of a soil's ability to supply nitrogen. Has been shown to be very sensitive to management, so a good soil health indicator, and also positive correlations with non-legume crop yield. As agronomists, figuring out drivers of crop yield is our holy grail, so this seems like a useful indicator for that. One of the practical research questions, I worked with Bob a couple years ago to develop this model of, can we replace this costly soil protein measurement with prediction to improve the cost effectiveness of soil health assessment, still provide the information, but provide them a model, estimated value. So with this kind of work, we build models, we validate models. We can use machine learning models to actually gain insights to what the variable is, in additional to just normal linear regression. And we wanted to demonstrate whether the model could pick up the same sensitivity to the measurement. So we have a very large national database of soil health measurements, one of the largest around, and about 4,600 samples. And the first process was determining the variables that were most predictive or most useful. We didn't want to include variables that weren't parsimonious, et cetera. So we removed a bunch. We have this great NAPSHM data set looking at a number of long-term experiments across the United States to be as our independent validation data set. We looked at a bunch of variable importance and partial dependence to gain insights, and we determined the sensitivity of measured and modeled soil protein on 57 long-term experiments. So this is kind of boring, but is useful for lab people who are interested in this. Basically, our machine learning model, we used one called random forest, is able to reduce the prediction error significantly compared to these other models. And we've determined that it's an acceptable RMSE, or root mean square error, basically prediction accuracy for commercial use. I'm going to skip this. And basically, we wanted to assess the sensitivity of the measured and modeled protein to management. We have 57 replicated long-term experiments, basically the best of long-term experiments across the country. Soil protein was sensitive to single way treatment effect at 36 out of 57 of those. Compare that to soil organic carbons, maybe slight greater sensitivity compared to soil organic carbon. We found that tillage and organic amendments were the management levers that were most likely to affect soil protein compared to cover cropping system, residue return, and cover crops. Found that soil protein was more sensitive to management at coarse textured sites compared to medium and fine textured sites. And that what was really cool at the end was seeing that my machine learning model was able to replicate those significant findings at 92% of those sites. So that's another really good proof of concept that our model is doing really well at estimating soil protein. So finally, let's go deeper. Soil health is really about usually, not because we're lazy, but we focus on the top 6 inches for practical purposes, but we really need to go deeper in understanding the effects of management, especially at our long-term sites. So what about the effects below 15 centimeters or 6 inches? And this is the focus of my chapter 5 on effects of tillage, cropping system, and soil texture on carbon stocks in 3 long-term experiments. So for context, soils are the largest terrestrial carbon sink and have a role to play in climate change mitigation. Notice how I said role. I think there's a lot of hype in the public about the benefits of carbon farming and the promises of regenerative agriculture in popular culture. So I think we need to be real. There's a lot of claims out there that regenerative agriculture is going to stop the rise of CO2 in the atmosphere. Probably not. And we've had a hyper focus on carbon and methane, but we can't forget about nitrous oxides that are really lower hanging fruit for addressing greenhouse gases from agriculture. So some bigger context. This was a optimistic soil carbon sequestration claim from some top leading soil scientists in this area. With large investments, potentially we can do 36% of total U.S. ag emissions, or 3.7% of total U.S. emissions. So some of the most optimistic soil scientists are very different from what we see in the popular culture sometimes. And then I also wanted to share some about the benefits of no-till and strip-till, because the results I'm going to present, I don't want to leave you with the wrong ideas at the end. So no-till and strip-till is incredibly important at reducing erosion, increasing infiltration by having residue armor the soil surface, really improves soil health and climate resiliency at the surface, significant improvement in operation profitability. The reason no-till and strip-till are so widespread across the landscape, partially because of the environmental benefit, but also because the economics of it worked out as well. May increase yields, and the context really depends on how well no-till or strip-till is implemented. So if you asked a soil scientist 20 years ago, does no-till sequester carbon? I think the mainstream view would be that it does. But is it true, and how relevant is it for New York State? So here's our what I'm calling plow till. This moldboard plow would be followed by secondary tillage. And here's a really innovative form of no-till, planting green into high residue biomass. So no-till increases soil organic carbon and soil health at the surface, but research has really challenged whether that holds for the whole soil profile. And it makes sense as soil scientists that we would think that what we're seeing at the surface probably holds true if you go down in depth, but it turned out that that wasn't true. So researchers began noting that basically a lot of these studies were done with less than or equal to 30 centimeters of sampling depth and were insufficient. And then when you go a little bit deeper, the data can tell you the opposite story, or it tells you that there's no differences. So these were some of these first papers about 15 years ago that began questioning this, and this has become kind of a main more mainstream view actually in soil science. But it's really challenging when we're communicating to farmers and then our stories are changing from decade to decade. So for my research questions, I was asking how much organic carbon has been lost for some of these long-term experiments, looking at some of the 3 longest term tillage experiments in New York. What does no-till lead to greater soil organic carbon stocks compared to plow-till in New York State? We need to ask the question, do our long-term experiments, what role does texture play, and are there interactions between tillage and cropping system, organic carbon input level on soil organic carbon stocks? So here's our long-term tillage sites. These were basically started at the early part of Harold's career in 1992. Spent a lot of time 30 minutes south of Plattsburgh for my research. And at the Willsboro Research Farm, great farm manager up there, Mike Davis. And we have basically these 2 parallel experiments. Really great site because we can drive less than a mile and be on a clay loam site, one of the clayest soil textures I work with, and then also a loamy fine sand site, which is one of the coarsest textures, kind of like a beach. And we have this contrast of plow-till, no-till corn silage. You can see that greater than 90, maybe 95% of that above ground biomass is harvested, which is a big issue in corn silage systems. And these systems are also unique in that there are no manure inputs here. We also have a 3rd experiment at the Musgrave Research Farm looking at plow-till versus no-till corn grain. So that maybe 50 to 60% of those, 55% of those above ground biomass is going back into the soil. And we also had the introduction of cover crops at the last 7 years of the experiment. This wonderful guy named Matt Ryan came to Cornell and asked Harold if he could add some interseeded cover crops to the experiment. So for methods, pretty straightforward. I collected 5 0 to 45 centimeter cores per plot, 2 adjacent cores on sod ground to understand the effects of management on soil health relative to under adjacent sod. So 270 cores altogether, about 90 cores each year. Split cores into 0 to 7.5 centimeters, 7.5 to 15, 15 to 30, 30 to 45. Measured bulk density, submitted samples for total carbon, nitrogen, soil inorganic carbon. Organic carbon is total carbon minus inorganic carbon. Here's some really cool pictures. You can really see the stratification of carbon in the no-till system versus the deeper mixing in plow-till. So how much soil organic carbon has been lost since the initial conversion of hay ground to this continuous corn silage system? And the Willsboro plots give us this unique opportunity because those before the corn silage experiments, they were in long-term sod for decades before that. So we can really assess what that decline's been due to this intensive corn silage system. Significance, we kind of mentioned that last week in Allison's talk and the questions, but corn dairy cropping rotations are changing nationally in the Midwest and here as well, where we're seeing alfalfa acreage go down. In New York, that's about 40% or 345,000 acres in the last 20 years. And some of that decline is being made up with more intense dairy cropping rotations with more corn silage, basically, mostly driven by economic factors, but has negative implications for soil health. Basically, we used the sod adjacent benchmarks, and we also had some historical samples from 1995 from the early days of GPS that we were able to use to estimate initial carbon stocks. So here's my estimate of initial carbon stocks for the loamy sand and clay loam at 84 and 108. And now the current SOC stock from all the work measuring it, 52.5 and 76.6. Interestingly, both of these experiments, loamy sand and clay loam, lost a similar magnitude of carbon. But when you compare that to the initial stock, the loss is about a little bit higher of percentage of that initial stock for the loamy sand. So the big question from this chapter, at least, does no-till lead to consistent differences in carbon stocks compared to plow-till in New York? And no, only 1 of 3 sites had significantly higher stocks than our no-till. Loamy sand was almost significant. And then there was no difference, and actually a higher numerical value for the plow till. And this is really counterintuitive from being an agronomist and being around all of the excitement. Actually, in the clay loam site, you can really see the organic matter differences at the surface. It's about 1% difference if you measure the top 6 inches. So it's really interesting to see this. We can calculate the sequestration rates, which is basically the difference between them over the amount of years that the experiment was run. And we can see that the loam is the highest, and makes sense, -1. And these texture effects, at least for temperate climates, are common, that we see that the increases in carbon stocks under no-till are least likely in these heavier clay soils. That's something that I found a couple different times in other side-by-side experiments with differing textures. So why does no-till not lead to consistent differences in carbon stocks? Let's look at why. We have this very intense stratification of carbon at the surface, but plow-till has greater carbon in the plow layer and below the plow layer that makes up for those differences. So when we were just sampling to 30 centimeters or even 15, 20 centimeters, you see that huge no-till sequestration story. But here at the clay loam, where there was absolutely no differences, big difference at the surface, but was pretty much made up at 6 to 12 inches, and then slightly more deeper down. For the loamy sand, it's similar, except there's probably more movement of carbon through the profile because there's more larger pore spaces. But same story, no-till, huge difference at the surface, and then it makes it up at 12 to 18 inches. We see, which is similar to the literature, much greater carbon stratification in no-till grain experiments. The majority of the carbon inputs are coming from corn stover, whereas the majority of inputs from a corn silage system actually are coming from the roots. So it makes sense that there's less stratification in a corn silage system. We had some methodological challenges measuring carbon stocks at the Musgrave farm. So know what you're getting into if you're thinking about doing some carbon stock work at Musgrave. There's a lot of inorganic carbon or carbonates at depth in the B horizon and deeper in the soil. Makes sense. We know these are high lime soils, why there are productive alpha soils. Honeoy is our state soil. But I was really surprised that when you go down 9 to 12 inches, 12 to 16 inches, we had about 40 to 70% of our total carbon being inorganic carbon. So that creates challenges in terms of capturing what the organic carbon story is, which is what we're really after here. So we had cover crops drilled in the experiment. They led to some marginal increases at the surface, but the remaining cover crop effects had significant interactions with the tillage system. That means something happened. There was interaction between the tillage and the cover crops. And this is unique. I don't want to, it's an important finding, I think, that should be tested with further research, but basically we found an interesting signature that the plowing down of cover crops actually reduced the carbon in the plow layer. And one hypothesis of why that could be happening is positive priming, kind of where carbon inputs enable microbes to jumpstart existing organic matter in the profile. So some key takeaway messages: no-till consistently has greater carbon stocks at the surface, but this doesn't necessarily translate to greater soil organic carbon. Across sites, cover crops lead to marginal increases at the surface. And at one set, we saw this interaction, which I think can be tested in future long-term corn grain experiments. And I also think we really need to emphasis on carbon inputs as a mechanism. We've tended to emphasize this tillage mechanism, but I think we've need more emphasis on carbon input side of the story. And this framework, this research can really help inform sampling and interpretation of new carbon credit and emphasis in New York. We're going to be having a lot of carbon stock data being generated in the future due to this emphasis, and we need a way to be able to interpret it. And we need to maybe question why people are going down so deep. So bringing it all together, soil health and carbon sequestration, this triple threat to agriculture requires this emphasis on sustainable soil management. Increasing carbon inputs, reducing disturbance, always the same story. Good story. Soil health assessment and benchmarks can help us inform management and policy. Predictive models can help improve the cost effectiveness, so we can have a very comprehensive soil health test, but with less cost. No-till has consistent soil health, climate resiliency, and economic benefits, so it's really important to remember that. But it might not lead to this carbon stock story, might not lead to consistent differences in carbon sequestration in temperate climates. I think there's a lot of evidence that in drier climates and more tropical climates might have a bigger role. Need more emphasis on carbon inputs. And as researchers, we need to tell folks a story. Don't assume that the carbon stock or the soil health trends that you see at the topsoil are going to hold true when you go deeper into soil. So we're going to need more work that goes deeper, but probably not too deep. Luckily, we're not in the Midwest where you have to go down a meter. So that's a good news. So I want to thank my committee, Harold, for giving me space and direction over the last 5.5 years to become scientist I am. Johannes Lehman, Matt Ryan, support from the committee. Bob Schindelbeck, who has been really important in all of this work, is the author on all of these chapters. So consider him part of my committee as well. Great New York Soil Health team, great lab group, Mike Davis at the Willsboro Farm, a lot of undergraduate help, and then everyone from the Cornell Soil Health Lab, really appreciative of everyone. So thank you, and I can take some questions.
Thank you, Joe. It was fantastic. Congratulations to this great set of works and well presented. I was wondering, I wanted to go back to this dairy cropping systems, and you saw that you had sort of intermediate soil carbon levels between the cereal grain cropping systems and the veg cropping systems. And I was wondering, you said something about that you would assume that the dairy farming systems have a more complete removal of above ground biomass as fodder, but yet it was still higher. Is there an opportunity to ask questions about what the quality of input, especially later on you said we need to have more emphasis on quantity, but is this maybe an opportunity to ask questions about the quality of input, which dairy manure input would be very different from crop residue input?
Yeah, it would be interesting to have an experiment where I had a lot of manure, but that obviously can change then. At the Cornell Dairy Farm, you see incredibly high stocks from manure, but carbon inputs, I think, is a big part of it, but also the quality is huge in some of the Penn State work that I've been a part of, finding that although significantly lower biomass for crimson clover, for example, than a grass cover crop, seeing influences on how that's driving soil carbon fractions. So that it could, I think it'd be the same story, whether it's crimson clover or manure, could have similar
exactly.
dynamics.
And triggering different carbon use efficiencies where each is getting into that we get more carbon accrual due to switching on different microbial pathways that result in different carbon use efficiencies that are maybe with material that have lower CN ratios is a higher carbon use.
Yeah, definitely. I think it's important.
Great, thank you.
Yep. Yep, Yishi.
A wonderful talk.
Thank you.
And it's really, you are you are expert on the soil carbon in state of New York. And so also same issue about the cropping system. And then you showed that in the orchard actually have very low soil. And then what's the reason?
Debbie is actually going to do a whole presentation on orchard soil health in a couple of weeks, so that will be answered. But basically, there's a huge degree. There's some very healthy orchards and then degraded. But conventional orchard management is to leave basically bare strip, so no carbon inputs, little life, to create this non-competitive zone for the trees. So that's potentially one important reason.
Can I ask one?
Yeah, sure.
Question here? So you showed the tillage and the non-tillage comparing, and then your tillage basically increase the carbon in deeper layers, in especially clay. Is that due to the changing the rooting zone or rooting depths, or because of the vertical carbon movement down to the below the faulting layer?
Yeah, and I think there's a lot of factors. The most simple factor is you're just incorporating it deeper in the soil profile. So those inputs are getting spread out. But yeah, and I mean, there's a lot of potential interactions with soil structure and compaction that could be driving things as well.
Thanks for the presentation. So I was wondering, so there's some different work approach if we are to prescribe noting or stocking more carbon, because when you consider it higher, and this would be very useful for carbon credits and markets. I was wondering if this holds true also if you're just looking at the productivity of the soil, all the other advantages of soil health.
Yeah. No, I think there's tremendous reason to do no-till, but maybe it's not going to lead to, by itself, not going to lead to the carbon sequestration part. But huge benefits for surface resiliency, increasing infiltration, and reducing production costs. A lot of farmers think about the savings with no-till help them spend the extra 20, 25 bucks an acre to do cover crops. So there's so many reasons to do no-till, but I think we really need to emphasize this carbon input part. I think one of the reasons that we might have seen a difference at the no-till, the grain experiment, is potentially no-till mediated higher yields of the corn crop leading to the carbon stock differences. So that would actually be my best hypothesis for that one. So I think we need to think about the inputs telling a bigger part of the story. Sorry, Beirut.
People are great presentation of Amsili. So my question is the value of the transfer function that you developed for protection soil protein. I wonder what you use magnesium for predicting that, but not other catalysts. Is there any idea about that?
No, I did it I did it based on the individual influence of each predictor variable, whether it's reducing the RMSE or helping to increase the fit. But yeah, no, so that one was based on what I had access to, what made sense for maybe capturing soil type effects as well. Yeah, no, so it was.
Well, maybe you have a process-based approach for choosing the inputs parameters.
I ran a lot of different models and kind of did an additive step-by-step addition and subtraction of different predictor variables to see what the.
I'm not sure how could be this idea could be related with that. Magnesium bonds to pigments and pigment bonds to proteins. It could be from plants.
Interesting. Oh yeah.
The idea of,
I didn't think of that. We need the soil chemist to help me think of that. Yeah. Sorry.
This is a great talk. Actually, your chapter 5 is very similar with what I'm doing right now. So you found the carbon top and you found the carbon increase soil layer, carbon in the top soil layer. And how about in the deep soil layer? That decrease or very similar?
Yeah, this is just one experiment, so I don't want everyone to jump on that result. I think it is a possible mechanism. It was interesting to see that there was that interaction where it didn't happen in the no-till system, but it happened in the plow-till system. But yeah, I don't have Dana.
The research that you did is remarkable and raises many interesting questions. My primary question is, with the model that you're using and predictions that you could potentially sequester roughly 3.8%, I think, of what we need to sequester. Did that surprise anyone? Is that high or low? Is that reason for joy, or is that good or bad?
No, I mean, I think that's just what an estimate is for what agriculture's capacity is. I think we need to also focus on all the other important climate mitigation strategies and energy.
Was there hope for higher result?
No, I don't think necessarily. I think there's one thing that's challenging in the public, there's a lot of kind of overhype that we could be promising that this is going to solve the problem when it maybe is going to play a smaller role than we think.
Thank you.
So let's give George.
This has been a production of Cornell University, on the web at cornell.edu.